Good morning, everyone. We're from Group 10, and our presentation today is going to be on in vitro fertilization and intracytoplasmic sperm injection. These are some of the topics that we are going to be covering in today's presentation. And these are the group members of Group 10.
Assisted reproductive technology, by CDC definitions, are any fertility-related treatments in which eggs or embryo are manipulated. In vitro means performed or taking place in a test tube and culture dish, or basically something that is outside of a living organism. The oocytes mature in vivo in the ovary, and the embryos develop into pregnancy in the uterus. But the oocytes are fertilized in the petri dish. So briefly, here are some of the process of the in vitro fertilization. First, the superovulation of female is induced by hormonal stimulation. The collected oocytes are then fertilized by culturing them with the sperm in vitro. And then, the embryos are transferred into the genital tract of the female, and the embryo then develops in the uterus of the dam.
Now it's a time to play. The game is to guess what animals were born according to the years shown after this. Game starts now. Are you ready, guys? So, what animal was born first through IVF in 1955? It's a rabbit. Next animal was born in 1963. Hamster. For this one, let's make a quick guess, guys. Come on. Yes, you are correct. It's a mice. You know what animal is this? It's a horse. Okay, let's try for this one. It's a cattle. How about this? Do you guys know what animal is this? It's a goat. How about this one? Nice try. How about this one? Yes, you are right. It's a dog. May I have your attention, please? Listen to the sound and guess what animal is this? Here is the answer. If you are interested to know more, you can refer to these slides for more info.
Moving on to the procedure of IVF. The first step is dominant follicles need to be removed first via transvaginal recovery with an ultrasound-guided needle to allow other follicles to grow. Next step is the superovulation, meaning that FSH is injected to stimulate the ovaries to produce more oocytes. Then, the third step is the ovum pickup, which is to collect the oocytes from the ovaries. The fourth step is the maturation process. The collected oocytes are then placed in an incubator to mature them for 20 to 34 hours. Then, the mature oocytes and sperm are put together in a petri dish for them to fertilize with each other and co-incubated overnight. After that, the resulting embryo are matured for about a week in the lab by putting them in a series of media that mimic the reproductive environment to support the embryo to grow into blastocysts. Lastly, once it forms blastocysts, it is ready to be transferred to the animals, who can also protein it for later transfer.
Moving on to the indications for IVF. Firstly, to overcome physiological problems, IVF can be used to produce offspring from elite animals that are non-productive in traditional methods. Secondly, to preserve valuable genetics. In the case of the unexpected death or slaughter of a valuable animal, eggs can be rescued from the ovaries and the resultant embryos can be transferred into recipients. Next, IVF reduces cost of semen. As fertilization takes place in a microscopic environment, significantly less semen is needed to fertilize the same number of eggs. This is highly beneficial when a rare, expensive, or sex semen sample is used. Lastly, less hormone reliance. Growing evidence supports moving away from administering expensive superovulatory drugs, as maturation can successfully be performed in vitro. Therefore, donor animals are not exposed to any potentially harmful side effects due to these hormones.
These are a few drawbacks of IVF. Adequate facilities and equipment are needed for oocyte pickup, and these can be very costly to the farmers. Next, suitable temperature and pH are needed to mimic the animal's uterine environment in order to keep the oocytes viable. Hence, damage of oocytes may occur due to the manipulation done. IVF may also cause physical impact on the ovaries, as follicles aspiration is an invasive procedure which may cause bleeding or infection. And lastly, not all donors will produce viable embryos, and this could cause further economical loss to the farmers.
Since IVF is used to treat infertility in female, intracytoplasmic sperm injection, also known as ICSI, is used as an assisted reproductive technique to treat infertility in male. What is ICSI? ICSI is an in vitro technique in which a single sperm is microinjected into the cytoplasm of a mature oocyte to overcome infertility. For equine, ICSI is used to reproduce early individuals. Failure of superovulation and conventional IVF have been the main reason for the use of this technology in horses. For swine, ICSI has been successfully used to produce transgenic animals. In pigs, IVF leads to high frequency of polyspermy, causing chromosomal abnormalities in embryos. For cattle, the use of ICSI for high genetic value cattle to maximize semen utilization and shorten generational interval. However, in cattle, low success rate reported due to failures of chromatin decondensation and oocyte activation.
Now, let's guess what animals was first studied for ICSI in 1914. It is starfish. Now, let's guess the next animal that being studied and reported. These are sea urchin and amphibians. In 1976, can you guess what mammals will succeed in fertilization through ICSI? Yes, it's a hamster. In 1990, kill bovine spermatozoa were microinjected into bovine oocytes. Moving on to 2003, tail-cut spermatozoa were microinjected into the ooplasm of goat and pig oocytes using a piezo micropipette driving system. And lastly, in 2012, ICSI also was reported in horses.
Next, I will talk about the intracytoplasmic sperm injection procedure. So before I talk about this procedure, I want to introduce an important apparatus, which is the inverted microscope. So this inverted microscope is coupled with micro manipulation system, and other than that, it also comes with injection pipette and also the holding pipette. So both of these pipette will be used to hold the sperm and also the egg later. So now let's move on to the procedure. Firstly, the sperm cell will be selected from the frozen thought semen and placed into the PVP droplet. This is important to reduce its motility. Then the tail of the sperm will be cut with the circular motion of the injection pipette, and a single sperm cell will be sucked into the injection pipette. And in this injection pipette, the head of the sperm will be facing towards the tip of the pipette, and the tail will be on the opposite side. Okay, this is important to ensure that the head of the sperm will penetrate the egg first. Okay, next, the mature egg or the oocyte together with its polar body will be held by the holding pipette, and then a small amount of ooplasm from this oocyte will be aspirated into the injection pipette. After that, the sperm cell will be injected into the oocyte, and once the sperm cell injected into the oocyte, the injection pipette will be removed. After that, the oocyte will be activated and start to divide and undergo cleavage. And finally, after this process have finished, it will proceed to be cultured in the species-specific medium before transferred into the female uterus.
So let's look at the indications of ICSI. It is recommended when other reproduction techniques fail. It is performed when there is male factor infertility caused by abnormal sperm production, blockage of delivery of sperm, low sperm production, or ejaculatory disorders. ICSI also allows for sperm sorting, preservation of valuable genetics, production of transgenic animals, and conservation of endangered species.
So for advantages and disadvantages of ICSI. For advantages, It is used for animals with severe infertility, and it is an alternative when IVF is not effective. Only few sperm is required for the procedures, and it allows selection of good sperm, thus it increases fertilization rate. However, for disadvantages, it is more invasive. Sterilization can damage the oocyte, and damaged egg will be degenerated. There is also high risk of producing unhealthy offspring. And lastly, the gene that causes infertility will be inherited to the offspring.
All in all, this is the difference between IVF and ICSI.
Here is the list of our references. Thank you so much for listening, and we hope that you have enjoyed our presentation.